Glycogen is a polymer of glucose molecules. It has a large size and a highly branched structure. In glycogen, glucose molecules are linked to each other by alpha 1,4 glycosidic linkages, and branching arise by alpha 1,6 glycosidic bonds at about every 10th residue. Glycogen occurs in the cytosol of the cells as granules, which also contain the enzymes that catalyze its formation and use. Muscle and liver are the major sites for the storage of glycogen. Although its concentration in the liver is high, the much greater mass of skeletal muscle stores a greater total amount of glycogen. Liver can mobilize its glycogen for the release of glucose to the rest of the body, but the muscles use it only for its own energy purpose. Glycogen is synthesized from glucose 6 phosphate. Glucose 6 phosphate is first converted into glucose 1 phosphate by the enzyme phosphoglucomutase. Glucose 1 phosphate is then converted into an activated form, UDP glucose, that is uridine diphosphate glucose, which can be incorporated into glycogen. UDP glucose is synthesized from glucose 1 phosphate and uridine triphosphate, and this reaction is catalyzed by the enzyme UDP glucose pyrophosphorylase. In the reaction, the carbon 1 of the glucose 1 phosphate is esterified to the diphosphate moiety of UDP. UDP glucose is then added to the non-reducing terminal residues of glycogen. It is transferred to the hydroxyl group at carbon 4 terminus of glycogen to form an alpha 1,4 glycosidic linkage. In the process, UDP is displaced by the terminal hydroxyl group of the growing glycogen molecule. The reaction is catalyzed by glycogen synthase, which is a key regulatory enzyme in glycogen synthesis. But glycogen synthase can add glucosyl residues only if the polysaccharide chain already contains more than 4 residues. Thus, glycogen synthesis requires a primer. This role is played by a protein called glycogenin. Glycogenin is composed of 2 identical subunits, each bearing an oligosaccharide made up of few glucose units linked by alpha 1,4 glycosidic linkage. Carbon 1 of the first unit of this chain, the reducing end, is covalently attached to the specific tyrosine in each glycogenin subunit. Each subunit of glycogenin then catalyzes the addition of few glucose units to its partner in the glycogenin dimer. Here also, UDP glucose is the donor. After the synthesis of oligosaccharide, that is primer, glycogen synthase takes over the role to extend the glycogen molecule. Glycogen synthase catalyzes only alpha 1,4 glycosidic bond formation. Branching in glycogen molecule is done by a separate enzyme called branching enzyme. This enzyme is also known as amylo 1,4 to 1,6 transglycosylase. The branching enzyme moves a 7-unit segment of alpha 1,4 residues from a glycogen chain to a carbon 6 hydroxyl group of a glucosyl residue that is at least 4 residues away from an existing branch. This is clearly visible in the diagram. Branching is necessary in glycogen because it increases the solubility of the molecule.